Hello students, the session will start at 3 p.m. sharp. Let's wait for some other course students to join. I know there are a lot of students enrolled in this course, so I'll carefully admit them, whoever is requesting me for the call joining. We'll wait for 5 more minutes and then start our session. Hello, students. I think we have a considerable amount of attendees now. I'll keep admitting others to this call upon request. It's time we start this session. Let me first start by introducing myself. My name is Ashutosh Behera, and I'm a PhD student, PMRF scholar at IIT Kanpur in the Department of Biological Sciences and Bioengineering. And I am the registered TA, teaching assistant, for this course, Nanotechnology in Agriculture. And this course is taken by Professor Mainak Das, and you guys must be familiar with this course since week 3 lectures are already out, and I hope you are following through your lectures honestly. And regarding the assignments, I am here to help you with the assignments. So what we'll be doing in this session is we will try to solve an exemplar assignment. So I will not help you with your original assignment that you have to do yourself for your exams, but I'll help you with a previous year or similar kind of questions, exemplar assignment, and we will see how the questions need to be solved and understand the concept behind these questions, which will help you to solve your assignments of week 1, week 2, week 3, and following in the course. Is my voice clear? Can anybody confirm? Yes. So before starting these sessions, I want to establish some rules we will follow throughout this course and these TA live sessions. So all the participants require to mute themselves and keep their video off during the whole session. And as host, I will enable and disable them on the basis of their questions. So whatever interaction, this session will be interactive session. I'll be asking you questions and we will be solving the questions together, concepts together. So whatever interaction you and me will have is through the chat box. So if you have any queries and doubts, you can post it in the comment box, which I can take a look at the end of my presentation. So I'll solve 10 sample questions for you today, and then we will see how these are solved. Then I'll take up your questions on these on how to solve on the subject after these sessions. So during that, please mute yourselves and do not open your video chat. So I think there are 60 students joined till now, and it's 3-5. I'll be admitting other students who are requesting to join, and with that much of an introduction, I'll share my slides. You you can just confirm whether my slides are visible. See, all the participants are required to mute themselves and turn off their video chat. Wait, let me post it. Let me post it in the comment box here. Yeah. With that, I'll now start sharing my screen. Yeah, if is my screen visible? Can everybody see the slides? Please type yes or no in the comment box. Yeah, please communicate with me through the comment box. Is my slide visible? Can you see it changing? Just type yes or no if you just want to respond. Yes, fine. So let's start this session for practice assignment week 1. So this is a replica, or these are similar kinds of questions what you might be facing in your current assignments and the upcoming assignments. And for the brief introduction again, so the course that we are going through together is Nanotechnology in Agriculture by NPTEL, and Professor Mainak Das is the course instructor. Let me just switch on my pointer. Yes. So Professor Mainak Das is a course instructor for Nanotechnology in Agriculture, and I'll be your teaching assistant for this whole course. My name is Ashutosh Behera, and I'm a PhD scholar and PMRF from IIT Kanpur. So starting off with the first question, what is the main constituting element of graphene? So I'll read the question again, and you might also be able to see this question. What is the main constituting element of graphene? So there are four options: lithium, potassium, carbon, silicon. So I want you guys to guess the correct answer. Even if you are not correct, it's okay. You just guess according to the lectures and whatever your knowledge is. Just type in the comment box which option should be the correct option for the question. What is the main constituting element of graphene? We will collect some answers and then move forward as what is the correct answer, or as if you guys were able to guess it correctly or answer is correctly. So I'll wait for 5-10 seconds and let guys answer. So just mention in the comment box what could be the correct answer for this question. For people who have joined just now, we have already started. We have already started the problem solving session, and the first question we'll be taking on is what is the main constituting element of graphene. I request all the participants to switch off their mic and video chat during this whole session, and please type the answer in the comment box. What do you think is the constituting element of graphene? Yeah, as I can see, some people. So there are two school of thoughts. Some group of students say it's carbon. The other group of students say it's lithium. So carbon and lithium, right? For students who have joined just now, I request you all to switch off your mic and video chat and try to answer this question. So we have already received some answers. So to the students who answered carbon, I want to tell them that this is the right answer. It's not lithium, potassium, or silicon, but the main constituting element of graphene is carbon. So if you see, graphene is one peculiar allotrope of carbon. What do I mean when I say allotrope? So allotropes are different forms of the same element. So the elemental unit is carbon, but allotropes are different forms of the same element where the atoms combine in different ways so that at the same temperature and pressure they can exist in different forms. So it says that the same carbon atom can combine in different ways to produce various structure which are known as allotropes, and at the same temperature and pressure conditions the same carbon atom can exist in different forms. So and these different forms are known as allotropes. So graphene is one of the allotropes of carbon. And why is it called peculiar? Let's look at some of the major allotropes of carbon. Yeah, students, please, I request switch off, turn off your mics and your video chats. Any communication, please communicate it via the chat box. The first questions are has already been answered. The answer is carbon. So as I was saying, we will look at certain allotropic forms of carbon. Graphite is the first allotropic form. Another form is diamond, fullerene, and graphene. So all these are four different allotropic forms of carbon. So if you see the arrangement of carbon atoms, so all these blue atoms, these purplish blue atoms are carbon atoms, wherever my pointer is following, right? So a single carbon atom can exist in itself, but when a group of carbon atoms combine in different ways, they form allotropes. So here in graphite, we can see a hexagonal arrangement of carbon atoms like honeycombs, and these arrangements form a single layer, and many such layers are packed together with again interaction between carbon atoms, and such layers, multiple layers packing together, creates this structure, and this substance forms the graphite. Diamond, we all know, must have known that diamond is also a form of carbon which forms under high pressure and high temperature. So what happens in a diamond mine or under high temperature and pressure? Carbon atoms get compressed and they form unique lattice-like structures where these carbon atoms bond together or combine together with other carbon atoms to form a specific kind of arrangement, which forms this allotrope called diamond. And in fullerene, we see fullerene is also a allotrope in which carbon atoms are arranged in pentagonal rings and in size of a football. So if you look closely at fullerene, the carbon atoms on the surface are arranged in a spear kind of arrangement with pentagons, and the middle structure is a hexagonal structure resembling the football, and is also called Buckminster fullerene. So graphene is also one peculiar allotrope of carbon. Let's look at how graphene looks like. So if you pay close focus, this is the structural arrangement of graphene. So different carbon atoms are again assembled here in a hexagonal structure, and many such hexagons, hexagon number 1, 2, 3, 4, 5, 6. So we are looking here at six hexagonal structures in a single layer. So such many rows and many columns form this mesh-like structure, which is the arrangement for graphene. So it's the same carbon atom arranged differently, and graphene is also unique because carbon-carbon double bonds is present in graphene, which was not present in earlier graphite, diamond, and fullerene allotropic forms of carbon. So in graphene, we observe carbon-carbon double bonded structure. That's why we called it a peculiar allotrope of carbon. And this graphene forms a single layer, and this is the name it has been given: hexagonal honeycomb lattice. So this lattice structure is formed by hexagonal honeycomb-like structures where honeybees make their home. They have similar kind of structures forming hexagons. So to the students again who have recently just joined, please mute yourselves and mute your video chat. Any communication with me will happen via the comment box, and reserve your questions to the end of this session, where we'll try to answer the questions. So the first questions we have already encountered for the practice week assignment, and this will give you an idea to solve your own assignments for this course. So as we were talking again, structural arrangement of graphene, we have seen it looks like a hexagonal honeycomb lattice. Now if we have a graphene layer and there are multi layers of graphene, that makes a similar kind of arrangement of graphite. So this single layer of graphite resembles graphene a lot, but graphene has carbon-carbon double bonds. And this is how a graphene based nanomaterial looks like, since we are pursuing this course for nanotechnology in agriculture. So graphene carbon fiber is quite flexible and transparent. And here are some of the properties of graphene, which I have listed down for you. Graphene is the thinnest compound known to man at 1 atom thick, the lightest material known, with 1 square meter weighing around 0.77 milligrams. So this polycarbonated layered structure is the thinnest compound known to man at 1 atom thick. So if you take a single layer of graphene, it will not be more than 1 atom thick. And it's also the lightest material known, where only 1 square meter is weighing around 0.77 milligrams. It's not even 1 milligram. So it's very light. And that's why graphene is finding its way as a base material for nanotechnology, and many upcoming devices which use nanotechnology use graphene as a material for manufacturing. And also graphene is the strongest compound discovered, between 100 to 300 times stronger than the steel. So graphene is also extensively strong, very strong, stronger than steel, not just 1 or 2 times, 100 to 300 times. And it's also a good conductor of heat at room temperature, and also a good conductor of electricity. So it also conducts heat and electricity. So if you try to relate the structure of graphene and graphite, we all know graphite is also a good conductor of electricity. So graphite is nothing but similar kind of arrangement of multiple layers of graphene, but graphene has double bonds, and graphene is also a good conductor of heat and electricity. And other notable properties of graphene are its uniform absorption of light across visible and near infrared parts of the spectrum. So this statement says that graphene is able to absorb light from the visible spectrum, and also at the near infrared parts of the spectrum. So this is an background explanation for our first question, where we had what is the constituting element of graphene, which is carbon. So arrangement of carbon, it's an allotrope of carbon. Is the first question and clear to everybody? If yes, then please type yes in the comment box. Okay, so we are getting some responses. Very good. So yeah, I got it. Thank you, students. Yeah, let's move on to the next exemplar assignment question. So the next question that we are going to deal with is, what is the ideal pH of soil for agriculture? I'll repeat the question. What is the ideal pH of soil for agriculture? And the options are pH 1 to pH 2, pH 4 to 5, pH 6 to 7, and pH 9 to 10. So students, I want all of you to think of this answer. Even if you do not know, just guess it. What do you think is the ideal pH of soil for agriculture? I'll wait for a few minutes, few seconds, and then look at all your answers. Okay, so there are already answers. Surat says 3, Prashant says 6, 7, Rashmi 3, Narsimha 4 to 5, 6 to 7, Abhishek 6 to 7, 6 to 7, option 3, 3 option. Okay. Okay, so we have reached a consensus here. So most of the students are saying 6 to 7. Let's see what the correct answer is. Well done. It's actually 6 to 7. You have all answered it correctly. It's good to see that you all are interacting very nicely and trying to answer the questions. As for somebody who has asked in the chat box, where can I get the assignment answers? So the assignment answers will not be provided to you beforehand, because it's a course and you need to solve the assignment on your own. After you solve the assignment, I think the answers at the end will be given. I need to consult with the NPTEL team and just tell you. But this is what my duty here is, to guide you with the example questions which will help you. You will have similar kind of questions for this assignment, so I can help you with that. Yeah, let's see. Okay, there is a interesting comment on the chat box. The ideal pH may depend on the crop type. Yes, AK, you are correct. We will learn about what is pH and what crops require, and then we will understand. Yeah, then we'll understand why ideal pH is 6-7. So let's move on to the explanation. So for simplifying things, I have included a chart here for you, for all of you, to see what is pH and what kind of solutions we can map these pH into. Okay, I request all the students to please mute your mic and uh, switch off your video chat. Then we can proceed with the answers. Yes, I think we can move on. So here is a chart showing the color coding for pH 1 to pH 14. As we all know, pH is denotes the concentration of hydrogen ions. So power of hydrogen ions in the negative logarithmic scale is the pH. So if we start from 0 to 1 pH, battery acid and HCl secreted from the gastrointestinal tract in the stomach is pH 0 to 1. So this is very high acidic pH, where we have the battery acid has pH of 0 to 1 and as such as HCl lining. Lemon juice, vinegar comes around the pH of 2. Grapefruit, orange juice, soft drink pH 3. Tomato juice is pH 4. Acid rain, black coffee is pH 5. So the black coffee we drink, the lemon juice we take in have all acidic pH. And can somebody tell me what makes a pH more acidic, and what are the ions which make pH more acidic and pH more basic? You can type the answer in the comment box. What ions make pH more acidic, more towards 5, 4, 3, 2, 1, and what other ions will make the pH go towards 11, 12, 13, 14? Let me see in the comment box. Yes, 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 students have answered correctly. So the H plus ions make the pH more acidic. A solution which has more H plus ion make the uh, make the solution more acidic, and OH minus makes the solution more basic. So let's go into the scenario of our question. What pH is optimum for plant growth, right? So here is a table showing us what is the optimum pH and what are the characteristics of plant growth in different pH. So when we start from low pH, pH less than 4.6 is too acidic for most of the plants, while the pH 5.5 shows reduced soil microbial activity. So growth of the plants is also dependent on the surrounding microbes, which we will come further in a question where we have that microbes also help plants in many ways. So if the soil microbes are removed, then the plant health is affected. At acidic pH, many soil microbes activity are reduced. So the optimum pH here is mostly slightly acidic to near neutral pH, which is optimum for the plants. 6.0 to 7.5 is acceptable for most plants, which was our option 6 to 7. 6.8 to 7.2 is near neutral. Some plants also grow in this range. And pH 7.5, iron availability becomes a problem in alkaline soil. So when we are crossing pH 7.5, we are reaching towards the alkaline region of the pH scale. And then 2 alkaline for most of the plants is greater than pH 8.3 is 2 alkaline. But there are some exceptions in some plants which grow well in acidic and extreme alkaline pH. There are certain microbes also which grow in that crops in that pH region. So if we again try to review, So the ideal pH for plant growth is from 6, which is slightly acidic, to towards neutral. And when we reach to the acidic side below 5, below 6, there can be nutrient deficiencies and toxicities of metal ions like iron, manganese, and aluminium. While in highly alkaline media or the soil, we have nutrient deficiencies of carbonate, bicarbonate, and sodicity. Sodicity means high basicity, soda, deposition of soda. So there are many alkaline lakes, uh, especially where you find flamingos. So those are very alkaline lakes, and acidic areas where there is sulfur mines and deposition. So in acidic soil, we will find nutrient deficiencies for metal ions, obviously, because metal ions always conjugate with hydroxyl ions and form the alkaline and form the alkaline media. So in acidic media, we won't have more of the hydroxyl conjugated groups. So it's obvious that we will have iron, manganese, and aluminium deficiencies. And in highly alkaline medium, similarly, the opposite trend will follow. Acidic ions like carbonate, bicarbonate uh, will uh, will have nutrient deficiencies. And here is the answer to what a student was saying, that there are some species which are tolerant to highly alkaline media, and in some that can grow in highly alkaline media. And also by applying lime to the acidic soil, we can reduce the acidity and bring it to the near neutral range, which is ideal for plant crop growth. Is it clear to everybody? You can just let me know if it's clear, this question and the answer. You can just type yes in the chat box and let me know. We'll move on to the next question. Yes, great. Yeah. So the third question, what does the acronym NPK fertilizers stand for? I'll repeat, what does the acronym NPK fertilizers stand for? The options are nano pure krypton, nitrogen phosphorus potassium, nitrogen potassium krypton, nitrogen polonium chiromon. I'll just give you some time, 5-10 seconds, to think of the answer. You can just write the answer in the chat box. Nitrogen, phosphorus, potassium, nitrogen, potassium, krypton, nanopure krypton, nitrogen, polonium, chiromon. Okay, people have mentioned option 2. Yeah, so let's see. Yeah, you guys are correct. It's nitrogen, phosphorus, and potassium. And let's see what are these fertilizers containing of. So three main macronutrients needed for plant growth are these three elements consisting of nitrogen, phosphorus, and potassium. Nitrogen is seen for required for leaf growth. Phosphorus is involved in development of roots, flowers, seeds, fruit. Potassium for required for strong stem growth, movement of water in plants, promotion of flowering and fruiting. So these are some of the important roles played by nitrogen, phosphorus, and potassium. It doesn't mean that these are the only roles. They, being macronutrients, are involved in many functionalities of the plants. So what are macronutrients? Macronutrients are those nutrients present and consumed in larger quantities in the plant tissues, from 0.15% to 6% on a dry matter moisture basis. So if you take the dry weight of the plant, 0.15 to 6% of the plant would be constituted by macronutrients. and some of them are nitrogen, phosphorus, and potassium. And this is a schematic representation for NPK fertilizer from IFCO. So if I ask you, where do we think nitrogen can play a role? So all the proteins in the plant or in any living organism per se, all the proteins have major constituents of carbon, hydrogen, oxygen, and nitrogen. So nitrogen is needed for all the protein synthesis and transport. So water and sucrose transport across plants needs the presence of ions like potassium. Here we see that it's needed for movement of water in plants. So these ions create an osmotic gradient through which the water gets transported in the plant. And these macronutrients are also required in synthesis of the DNA. So whenever a plant cell is dividing or any cell is dividing, the DNA constitutes of two major components: nitrogenous bases, where we need nitrogen, and phosphodiester backbone, phosphodiester bonds, and uh, phosphate groups connected via phosphodiester bond, and the phosphate backbone of DNA needs phosphorus. So we see that NPK macronutrients are the essential components of plants which play a role in proteins, in transport, and also a major part of DNA. So these are some important functions. There can be other functions. There are other functions where these are involved. So, so these elements, macronutrients, are often supplied as fertilizer or supplements to the crops. So NPK stands for nitrogen, phosphorus, and potassium. Is everybody clear? You can please let me know. Just type a yes. Yes, yes, I'm getting responses. Okay, we have solved number three question. And for students who are posting updates and asking about other assignments and questions, see, this is an exemplar assignment. So this is not the actual assignment that you have been given to. And the answers and all, even I have little information when will you gain the answers. But what I can help you as a fellow student, as a fellow course taker and TA, to understand the concept and help you with solving your assignments. So for week 2, week 3, week 1, so this is none of your assigned assignments. This is a practice assignment. So we'll be practicing such assignments every week, which will be related to the lecture content of that week. So week 1, 2, 3, I suppose uh, these are the topics that have been covered. Questions are related to that. So without further delay, let's move on to the next question. So the next question here displays Haber-Bosch process was discovered for the synthesis of what? Ammonia, carbon, nitrogen, and phosphorus. So I'll repeat the question. Haber-Bosch process was discovered for the synthesis of what? Ammonia, carbon, nitrogen, phosphorus. See, these MCQs are all nice and easy questions. You just need to give a little bit of thought, and connect it to the lecture. I'll just give some time for students to respond. Please try, even if your guess is wrong, we will just reach to the correct answer and try to see what is the rationale behind the answer. You can type in the comment box. Haber-Bosch process was discovered for the synthesis of dash. Okay, some people have answered nitrogen, others have answered ammonia. Yes, yes, you guys are close. So the answer to this question, yes, option 1, ammonia. So it's not nitrogen. Haber-Bosch process was discovered for the synthesis of ammonia. So you are close. It's not carbon phosphorus. It's a nitrogen containing compound, but it's not nitrogen. So this is Fritz Haber who discovered this process, and he was a German scientist. who discovered an efficient way of producing ammonia from hydrogen and atmospheric nitrogen. So in this process, also known as Haber's reaction or Haber-Bosch process, atmospheric nitrogen and hydrogen are combined in presence of catalyst and high temperature pressure conditions to give ammonia. And it's also a widely accepted industrial process for synthesis of ammonia. But what makes it so special? What is so special about Haber's process? In Haber's process, we use atmospheric nitrogen. As you guys know, atmospheric nitrogen or any kind of nitrogen gas is an inert gas. Mind the different definition difference between an inert and a noble gas. Nitrogen is not a noble gas, but nitrogen is an inert gas. Noble gases are helium, neon, argon, and those noble elements. Nitrogen is not a noble gas, but nitrogen is an inert gas. What do I mean when I say it is inert gas? It has very low reactivity towards anything. So when we just have nitrogen in our air, it does not under normal condition it does not show much reactivity. But under certain pressurized conditions, pressurized means under certain conditions, nitrogen can react with the oxygen in the air. So atmospheric nitrogen reacts in the presence of lightning bolt with the oxygen in the air to form nitrates to form nitrogen dioxide, and which further go into the soil, form nitrates, and also some of the nitrogen dioxide and other oxides of nitrogen are formed, which cause acid rain. This is a this is a condition where an inert gas like nitrogen can also react during lightning strike. But readily atmospheric nitrogen, an inert gas. And preceding this discovery means before this discovery, reactions didn't involve use of atmospheric nitrogen as it was too difficult to make it react. So nobody before Haber could make atmospheric nitrogen react to produce anything. But Haber discovered this process of combining atmospheric nitrogen and hydrogen to produce ammonia. So this discovery was a breakthrough. It meant that ammonia could be produced efficiently and cost effectively. So efficient and cost effective. If you look at these two viewpoints, since atmospheric nitrogen is required and atmospheric nitrogen is very abundant in the atmosphere, so we just need to harvest those atmospheric nitrogen and combine with hydrogen, and ammonia can be readily produced for whatever quantity we need. So we will never have a loss of atmospheric nitrogen. So there is no need of acquiring nitrogen separately for this reaction. For ammonia, we can use atmospheric nitrogen and we have hydrogen, and then we combine it and we give ammonia. But for this discovery, Fritz Haber was also known as one of the greatest heroes and greatest villain. And why so? Because ammonia, the product which he generated from his reaction, ammonia is a starting chemical used in production of nitrate fertilizers. So many of the nitrate or nitrogen supplying fertilizers are made at the starting material is ammonia, and also ammonia is made for warfare explosives. Ammonia is also a starting chemical for warfare explosives like trinitrotoluene, nitroglycerin. So this process was a breakthrough in synthesizing ammonia or production of ammonia. As a science greatest hero, he could produce these ammonia can be used in nitrate fertilizers, but as a villain, these can be used also for synthesis of warfare explosives. So this is the history of Fritz Haber and how he discovered the the generation of ammonia from atmospheric nitrogen and hydrogen. And if you look at Haber's process, so here is a simplified schematic for the Haber's process reaction. So it's commercialized now, and many industries use this Haber's process to produce ammonia. So what we see here, if you follow my cursor, this is a simplified overview. Nitrogen and hydrogen are compressed in this compressor, and there are catalyst beds. So these catalyst beds consist of ferrous oxide or ferric oxide, generally ferrous oxide, so FeXOX, depending on the catalyst. And then through this catalyst beds, these compressed nitrogen and hydrogen is passed through, and there is a heater which generates a temperature of around 400 degrees Celsius, greater than 400 degrees Celsius, and the pressure inside this chamber is greater than 400 atmospheric pressure. So under these conditions, one molecule of nitrogen combines with three molecules of hydrogen in the presence of catalyst, ferrous oxide catalyst, at the temperature greater than 400 degrees Celsius and a pressure greater than 400 atmospheric pressures, to give two molecule of ammonia. And this is the whole reaction for Haber-Bosch process. So if you follow this reaction again, so catalyst beds, these compressed nitrogen and hydrogen will react at the catalyst beds, and this heater and this compressed chamber will provide the temperature and pressure required, and the ammonia generated here shown in the red arrow will go to the heat exchanger. Here whatever nitrogen and hydrogen have not reacted or have not combined will be recycled again for this reaction, and from the heat exchanger, so since it's at high heat 400 degrees Celsius greater than, this needs to be cooled down. Ammonia will then pass through this condenser where cold water is dispensed. So this green coil you see, cold water is always running through this green coil, spiraling again, and this pipe in this chamber, ammonia gas is passing through. So it will always come in contact with the cold coil of the cold water. So cold water will come through this pipe, and then when it comes in contact with ammonia, there is heat exchange. Now ammonia gas will little cool down in temperature, and the cold water will absorb the heat. Now hot water will go out through this. So this chamber is called the condenser chamber, and ammonia will then be collected from a refrigerated unit. And here also there is a bypass where unreacted nitrogen and hydrogen can be recycled to this heat exchanger and then follow the same path through this recycled N2H2 and enter the catalyst chamber where this Haber-Bosch process is occurring. So we can summarize I'll repeat it again. We can summarize the formation of ammonia where one molecule of nitrogen and three molecules of hydrogen combine in the presence of ferrous oxide or ferric oxide, greater than 400 degrees Celsius temperature and a pressure, high pressure at 400 atms gives two molecule of ammonia. So, so to repeat the answer again, Haber-Bosch process is a method discovered for synthesis of ammonia. So is it clear to students? Please just let me know by typing yes. Okay, I see some responses. Yes. So yeah, very good. So the next question has come up. Which of these is not a micronutrient for plants? This is the question. Which of these is not a micronutrient for plants? Options are iron, copper, nitrogen, and boron. Yeah, this question is kind of intuitive since we already had the previous questions on NPKs and others, macronutrients. So which of these is not a micronutrient for plants? I almost hinted the answer. The students just try to guess, even if you're not correct, it's okay. I'm here to help you for solving these questions. I'll just wait 5 seconds, let the students answer, and we'll move slowly. Okay, some people have answered iron. There are some other answers coming: nitrogen, boron. Okay, some more answers. So there are, for your information, there are almost 92 students now who have joined this session. So just go with me and pay attention to what we are discussing here. It might help you with your assignment solving. So most of the answers are now here. So there is a varied. response. Some people say iron, nitrogen, boron. So the correct answer to this question is nitrogen. Nitrogen is not a micronutrient for plants, because just in the previous question we had the whole information about how nitrogen, phosphorus, and potassium are macronutrients which are supplemented in the NPK fertilizer, right? So nitrogen is a macronutrient. It's not a micronutrient, while the other threes here, iron, copper, and boron, are micronutrients. So the answer will be option C, nitrogen. Now let's delve into what are micronutrients. So if you just correlate with the definition of macronutrients, which are present in 0.15 to 6% of the dry weight of the plant and present in a larger quantity and consumed also in a larger quantity. So micronutrients, on the contrary, are present in the plant tissue in quantities measured in ppm ranging from zero point 0.1 to 200 ppm, or less than 0.02% of dry weight. So you see here it's so less. It's less than 0.02% of the dry biomass, but still micronutrients are required. Plants need these nutrients to function properly for their proper health. Thereby, because of their low, because of their absolute requirement, but also less quantity of requirement, they are known as micronutrients. So the three micronutrients that we encountered in our question: iron, copper, and boron. So we will just look at what functions briefly. What functions do they do? So boron is believed to be involved in carbohydrate transport in plants. It also assists in metabolic regulation. Boron deficiency will often result in bird dieback. So the birds die if there is a boron deficiency, and it is involved in functions like carbohydrate transport in plants. And it's also assist in metabolic regulation. So many of these metals play part in metabolic regulation because the metabolites that are transported throughout the plants are known to harbor many of the metabolites. These transport occur through boron. So the next micronutrient that we are going to study, copper. Copper is a component of some enzymes, and symptoms of copper deficiency include browning of leaf tips and chlorosis. So copper is a metal ion required as a uh, cofactor in certain enzymes, and loss of copper or deficiency of copper makes chlorosis. The phenotype that we see is chlorosis. Chlorosis is yellowing of leaves or loss of chlorophyll pigment from the leaves. So if we have copper deficiency in our plants, we might see yellowing of the leaves. And iron, as we all know, is essential for chlorophyll synthesis, which is why an iron deficiency result in chlorosis. So iron is an essential element, metal present in chlorophyll, as all types of chlorophyll, and which is why iron deficiency results in chlorosis. So here also we can see yellowing of the leaves if there is an iron deficiency. And this is the schematic telling you all the micronutrients known which are required for the plant in trace amounts, and the plant can acquire it through their diet or the soil, not from photosynthesis. So these are copper, boron, molybdenum, cobalt, iron, manganese, chlorine, and zinc. So there are almost 6 to 8 micronutrients which the plant require in trace amount, but it's necessary. And three of them that we just encountered: boron, copper, and iron. So the answer to this will not be uh, iron, copper, or boron. These are micronutrients, but nitrogen will be the answer since it's the macronutrient. Okay. Now moving on to the next question. Which of these is an insecticide? Okay, so now we are talking agriculture. Which of these is an insecticide? Options: A. Endosulfan, B. Atrazine, C. Naphthalene acetic acid, and D. Butachlor EC. I'll just give you a minute of time for all the students who are joining and leaving and who are all already been here. Just try to answer this question. Try to think of what can be the answer. Which of these is an insecticide? If you might have heard some of these chemical names, or some of them were in news recently or earlier, might have come across. I'll just wait for some time and which of these is an insecticide? Okay, so we have some answers. Okay, the response has gone down. Now only few students are answering. See, do not hold back. Need not fear wrong answers. Just try to attempt the question. Even if your answer is wrong, we all will move to the explanation of why the correct answer is the correct answer. Right? Just try to respond. I'll just wait a few more seconds. Then we can. Okay, let's see what's the correct answer is. So many students have answered endosulfan, atrazine. So stuck between these two. Okay, some people have also answered naphthalene acetic acid. So the correct answer to this question is endosulfan. So before looking more at endosulfan, I want to I want to rule out why the other three options would not be the answer. So atrazine, some people have answered atrazine. So you are too close. Atrazine is not an insecticide as per se, but atrazine is a herbicide. So there is a difference between an insecticide, pesticide, and a herbicide. So insecticide and pesticide target all these bugs and flies and pests and insects which directly damage the plant. But herbicides and weedicides are and different class of chemicals which target herbs and weeds. So unwanted plants are targeted by atrazine. Butachlor EC is also a herbicide, and naphthalene acetic acid is a organic compound for digestive disorders. It's human. So endosulfan is the correct answer. Endosulfan is the insecticide. Atrazine and butachlor are herbicides. They are not insecticides. Yes, somebody has written endosulfan is banned. Yes, yes, we will come soon to it. That why what is endosulfan and why is it not used now? So this is the complex structure of endosulfan. You guys need not worry about these structures. It's just a chemical representation, just to show you these lines that do not have any annotation of atoms are carbon, and then we have chloride. So this is a heavily chlorinated compound, and here we have a sulfide group here, SO3. So this is what endosulfan looks like. And as somebody mentioned in the comments, let's see what endosulfan has information to offer. Endosulfan has been used in agriculture to control insect pests, including whiteflies, aphids, leafhoppers, and beetles and cabbage worms. So endosulfan is in paper a good insecticide and a pesticide. It kills off whiteflies, aphids, leafhoppers, Colorado potato beetles. We all know crops get destroyed by all these insects and pests who who have the numerical advantage over crops. They can reproduce very fast and in large numbers to colonize a whole a field in a time span of, let's say, 2 weeks or 3 weeks, and that will destroy the whole crop field and result in huge losses and shortage of food. So to combat these situations, endosulfan had been used for killing all these insects and pests. And what is its mode of action? Due to its unique mode of action, it's useful in resistant management. However, as it is not specific, it can negatively impact population of beneficial insects. So endosulfan is highly potent. By the mode of action, it means it can kill without fail. But what a big problem is, it is not specific. So it's not specific to the bad kind of insects and pests. So if I tell you, in a crop field, so there can be two kinds of organisms or insects. Let's narrow it down to insects. One kind of insects are all these white flies, aphids, leafhoppers, and beetles and worms and ball beetles. So what they will do, they will try to destroy the crops, or they will try to expand their colony at the expense of the crops. They will infect the crop seeds, the flowers, some other parts of the plant, and destroy the crop. While on the other hand, we have useful insects which pollinate, which also help in pollinating the crop. So many insects are in symbiosis or in relation, in mutual benefit relationship with the crop plants. As we have microbes also, there are many insects which are beneficial to the cropland. But what did endosulfan do? Endosulfan killed all kinds of insect, irrespective of whether they are beneficial to the crops or to detrimental to the crops. So it can negatively impact the population of beneficial insects. And see, the third point says it's moderately toxic to honeybees, and it is less toxic to bees than organophosphate insecticide. So see, we already have an example. Honeybees are the insects. Without them, we cannot even think of growing any kind of crop, let alone fruit trees or any kind of trees. Because honeybees are the major pollinators of many crop fields and many flowering plants and other ways of pollination. Honeybees are the major pollinators. So endosulfan has been shown to be moderately toxic to honeybees. So if we apply endosulfan on the fields, it might kill the honeybees. So there is no point in eradicating the insects and pests which will attack the plant, because now the plant won't even have any pollinating agent. So the crop will anyway die. So this is a image or a perspective image of endosulfan bottle and how it was packaged. Now we come why endosulfan is not used now. So if we summarize again, endosulfan is an organochlorine insecticide. So I showed you on the structure there are so many chlorines in the carbon backbone, which is a persistent organic pollutant. So it's a pollutant, doesn't wash away. And these are the trees, plantations where it is used: cashew nut, coconut, rubber plantation. It is a bioaccumulant. So this is a term that people have used in the newspapers when there was a news regarding the ban of endosulfan. So it's a bioaccumulant. I'll explain what is a bioaccumulant. So if we break down this term bioaccumulant, we will see. So bio means living and accumulant means something which accumulates in the living things. So this endosulfan is so notorious a chemical, it will accumulate in living things. So if we suppose we spray a crop field with endosulfan, the crops will intake the pesticide, and then insects will be gone. But then the crops uh, cannot excrete the endosulfan, or it's not endosulfan cannot go out of the system. Even if the crop wants it to go out of the system, it won't go. And then the crops that we consume, so these crops, let's say rice or peas or dal, we consume, the humans and consumers consume this crop. Now this endosulfur will enter a new system. It will enter the human system, or let's say herbivores, any cattle grazing on endosulfan treated plants. will intake that and accumulate in its body. So by bioaccumulant, we mean this chemical is not getting out of the system and it's accumulating in the organism. So here is a small representative schematic showing low levels of endosulfan here have been moved over through food chains and then accumulated in the organisms. So generation by generation also it accumulates in the organism and it doesn't get out of the system. And what are the effects of endosulfan? So these are some of the major toxic effects of endosulfan. It is neurotoxic to humans, destroys the integrity of nerve cells. So nerve cell structure and morphology is destroyed by endosulfan and is also an endocrine disruptor. So what do we mean by endocrine disruptor? It will destroy the signaling caused by endocrine system. Endocrine system. Okay. So I have a short question for you. Students, can you tell me what is endocrine system? What are the major things they do? You can type in the chat box. What do you know about endocrine system? Have you come across this term? Endocrine system. It is for a hint. It's one of the major operating systems in our body. Like we have respiratory system, excretory system, we have circulatory system. There is also something called endocrine system. Yes. So there are a couple of answers. Hormones released directly to the blood, control of bodily functions via hormones. Yeah, these two students are absolutely correct. So the endocrine system in our body secretes or functions through hormones. So hormones are certain molecules which can be of protein origin, carbohydrate or glycoprotein, which are secreted from and can function through long distances. So if a hormone, let's say, is secreted in the brain, it can go and act its effect in the kidney, even in the kidney or lower parts of the body. While signaling proteins and all, these travel to short distances. Hormone can travel long distances. I would request all the participants to please mute their mic, and while we are proceeding with the assignment, please please mute your mic. Yeah, as I was saying that endocrine system functions through hormone, and endocrine system is one of the major coordinating systems in our body. So one of the best example of hormones might be testosterone and estrogen. So testosterone and estrogen are responsible for male secondary sex characters and female secondary sex characters respectively. So during female development and pregnancy, estrogen and progesterone plays a big role, and for male secondary sexual characteristics, testosterone is there. So these are all hormones secreted by different endocrine glands present in the body. So coming back to endosulfan, endosulfan is an endocrine disruptor. So what we saw when application of endosulfan, it disrupts the endocrine system. It caused delayed reproductive development. So reproductive development means development of male and female sexual characters and organs and reproductive system. So those are controlled by hormones, and these endosulfan went and blocked that. So there is delayed reproductive development, late sexual maturity, and autism. So autism is also a behavioral and neuronal disorder of the brain, and these endosulfan cause all these detrimental effects in human. There is late sexual maturity, autism, and delayed reproductive development. Hence, there was a ban on endosulfan, as a student mentioned correctly. In 2011, Stockholm Convention on Persistent Organic Pollutants, so this is a meeting that was held, banned the production, distribution, and use of endosulfan in India. India has agreed to phase out use of endosulfan by 2017. So, back like more than 10 years back, 2011, the ban was there, imposed by Stockholm Convention on endosulfan. But still, there are some parts which use endosulfan illegally under the radar. But there were endosulfan was making, at a time during 2010, endosulfan was making headlines of newspapers every day. And this tragedy especially happened affected people of Kerala, where where newborns were seen with deformities, like you can see here, a child with large head or megaencephaly, and people with lots other deformities in their bones, in their postures, in their muscle development. So endosulfan was interfering with the endocrine system. What else could we expect? The phenotypes were to be severe, so the effects were to be severe. And this happened in Kerala and in southern parts of India, where they have lots of coconut and rubber plantation, because endosulfan was used as a pesticide in coconut rubber plantations. So children in Kerala and southern parts of India suffered a lot, and there was a ban in endosulfan. So endosulfan became a highly controversial agrichemical due to its toxicity and potential for bioaccumulation. See, if endosulfan would have been excreted out of the system, then it would not have been a problem. But the thing is, it's a bioaccumulant and it accumulates in the human system or in any organism. That's what makes it more dangerous and disrupts the endocrine system. So, because of its threat to human health and environment, global ban on manufacture and use of endosulfan was negotiated under Stockholm Convention. So, this is just repeating. Not just India, endosulfan was also banned globally. Yeah. So that's for the sixth question that we encountered. Which one of was an insecticide? Is it clear to the students? If yes, then please let me know in the chat. Okay, great. We have received some responses. Very good. Please keep interacting, students, whenever we are trying to solve the question. Anyway, I will give you the answers. We are going through the questions one by one. But just try to guess. There is no need to shy out. Even if your guess is wrong or whatever you know the information is inadequate, I'm here to give you more information of what the answer is. So just try. We have a few more questions. I think four more questions. So just try to answer. Try to participate in this discussion. So it will be both beneficial for you and me. So moving on to the next question. Number 7. What was DDT used for? I'll repeat. What was DDT used for? It's a similar kind of question, uh, but we have options here. Weedicide. Is it a pesticide? Both A and B. Is a weedicide and pesticide, or none of the above? It's a weedicide, pesticide, both A and B, none of the above. I'll just wait for a minute for you guys to answer. What was DDT used for? Is it a weedicide? Is it a pesticide? Or both A and B? Just wait for a few more. Okay, there are many answers now. So let's see what's the correct answer. The correct answer is pesticide. DDT is a well-known pesticide. So to the people who have answered DDT is a pesticide, they are correct. DDT is not a weedicide. So a pesticide, again, I want to emphasize, a pesticide is a chemical which kills the eggs and larvae of a pest or an insect or a nematode or a bug or whatever is disturbing the crop. While a weedicide is a chemical used to prevent the growth of unwanted plants or weeds. Weeds are those plants which are unwanted or which are not important to us. The crop plant is the one which is important to us. So weed may not always be parasitic. A weed can be any happy plant growing apart from the crop. But the weed, but the but what happens when we apply the nutrients? That will be divided in between the weed and a crop. So now the crop will not get the maximum nutrients, and the crop growth and yield will not be maximized. That's a weed. So if we have a weedicide, then weedicide will prevent the growth of weeds, while pesticide will kill the insects. So DDT is a pesticide. Is somebody asking, sir, can I ask one doubt? You can post it in the chat box. I'll see if I have time for it. What is the full form of DBT? Okay, Kanparthi. Kanaparthi, sorry. Narasimha. Sorry if I mispronounce your name. I was just going to talk about DDT in the next slide. So just wait. Your answer will be there on the slide. I have explanation about DDT. So again, to clarify the confusion about or the distinction about pesticides and weedicide and insecticide, I have made a definition here. Pesticides are the chemicals that are used to destroy and kill the pests. So pests are insects or any other organism which are affecting the plant directly. Those are pests. But a weed is not affecting the plant directly. A weed is just, taking a part of its nutrition. If we have unwanted plants in a crop field, then whatever nutrition or fertilizer or water intake, that will be divided with the weed and the crop plant. So the crop plant would not get 100%. The weed will take away 30 to 40% and the crop will get 60%. But the pests are what which will damage the plant directly in 100%. So example of pesticides are DDT and malathion. These are two examples. Weedicides are the chemicals that have the capability to destroy weeds growing near the crops are termed weedicides. So any weed or unwanted plant growing near the crop, these weedicides will kill it. An example are glyphosate and ethyl ester. These are two examples. So two examples each for pesticides and weedicides. And this is how we apply, the farmers apply pesticides and weedicides or any first fertilizer for that. There are sprays of pesticides and weedicides to eradicate. And as a student Narasimha asked, what is the full form of DDT? So here is it. It is dichloro diphenyl trichloroethane. I know it's a lot big and a complex name, but and it's not abbreviated exactly as it is seen here. If we partake it, dichloro is for the first D, diphenyl is for the second D, trichloroethane is for the T. So DDT stands for dichlorodiphenyl trichloroethane. So it's a very complex compound, commonly known as DDT, short form, is a colorless, tasteless, and almost odorless crystalline chemical compound. And it's also an organochloride. So what we saw, endosulfan was also an organochloride, and DDT is also an organochloride. And this is the structure of DDT. So if you follow the structure here, this DDT molecule has two benzene rings. These are benzene molecules, chlorobenzene rings. So dichloro diphenyl. So the benzene rings with OH group are called phenyl, or this is the phenyl group. So dichloro diphenyl. So these are two groups, phenyl chloro groups here. Dichloro diphenyl, trichloroethane. So this here depicts a trichloroethane. So if you imagine a carbon, if you imagine ethane molecule, ethane's okay. So can students tell me what is the chemical formula for ethane? How many carbons and how many hydrogen? Please type in the chat box. Ethane, quickly. It's okay if you are wrong, just try to attempt. Yes, somebody answer. C2H5, C2H4, CH3, CH3. Okay, you guys are close. The answer is CH3, CH3. So Tara is correct. C2H5 is the ethyl group. So you are close. Ethyl group means devoid of the hydrogen. So this can be treated as an ethyl group. C2H5 is the ethyl group, but ethane as a compound is C2H6. Each carbon atom linked to three hydrogen molecules, and the other the same. So C2H6 is the ethane molecule. C2H4, Sweta, C2H4, I'm sorry, but it's an alkene now. Ethane is an alkane. So trichloroethane, we see this junction contains a carbon atom. Here, all these ends represent carbon atoms. So one carbon atom here, the other carbon atom here. Here, all the three hydrogens are replaced by chlorine. So now it becomes trichloroethane. So trichloro for the three chlorines here and ethane. And then diphenyl. So these are two chlorophenyl groups. So it's easy to remember, but you need not remember. Just for the sake of explanation, how DDT looks like. So DDT does not occur naturally. DDT is not naturally occurring and is synthesized by consecutive Friedel-Crafts reaction between chloral and two equivalents of chlorobenzene. So this is the reaction which is carried out to synthesize DDT, and in presence of an acidic catalyst. So in industry, this is manufactured this way. And what is the mechanism of action of DDT? How does it act as a pesticide or an insecticide? So what do DDT do? DDT opens voltage sensitive sodium ion channels in neurons, causing them to fire spontaneously, which leads to spams and eventual death. So I know it's a lot of information for you guys, but we'll take it slow. Sodium ion channels, voltage sensitive sodium ion channels. So every organism, every pest here has neurons. All these pests, neurons have many ion channels, or ion channels are proteins which help in transport of ions inside the cell and outside of the cell. It's that it's simplified definition. Ion channel is a protein which will allow ions to transport inside and outside of the cell. So this is here a sodium ion channel, means the ion that transports, the channel allows sodium ion to transport inside and outside of the cell. Voltage sensitive, it means this ion channel is sensitive to voltage. So if there is a change in voltage, the channel might open. If there is another type of change in voltage, the channel might close. And this channel allows passage of sodium ions. So what does DDT does? DDT opens this voltage sensitive sodium ion channel. Means now DDT will bind to this ion channel in the neuron of the pest or whatever insect. Now it will cause it to open and keep it open always. So what will happen when the ion channel is always open? Now sodium ions will enter and move out always, causing them to fire spontaneously. So this movement of ions inside neurons. So what is the generation of signal and electricity in neurons are the passage of ions. So it's a balance between sodium and potassium ion which generates neuronal response or neuronal activity. So what this DDT will do? DDT will sit on the sodium ion channel. Now it will always open it or keep it constitutively on. It will always be on. Now sodium will go move in and out. That will cause the neuron to fire very spontaneously. So whenever a neuron is not needed to fire, even then it will fire. So there will be neuronal activity when it's not needed, which will lead to spams and eventual death. So what will happen if you imagine, if in our individual or in a human, if there are neurons firing all the time, so these neurons are controlling many of the actions. Here spams mean muscular spams. So our muscle contraction and relaxation, movement of hands and everything, or of our abdominal muscles, involuntary muscles, smooth muscles, everything is connected by neurons. So now if a neuron will fire spontaneously, it means all this activity will eventually occur all the time when we even do not need it. So that will lead to a condition called spasm, and then it will lead to eventual death, kind of a paralysis situation, or due to increased hyperactivity. And that's what causes death of pests when DDT is applied to them. So DDT will open this sodium ion channel, which will lead to continuous firing of the neurons, which will lead to span of the pest and then eventual death. Yes. So is it clear for this question that DDT is a pesticide? It's not a weedicide. So first we looked at what are the differences between what is the basic difference between weedicide and a pesticide, and how DDT looks like, what is the full form, and how does it act on the target cells. Is it clear to the students? Just post a yes or no in the comment box, or we can repeat it. Okay, we have some responses. So please keep interacting, students. We have maybe three more questions. So this was question number 7. Next question coming up is number 8. We'll solve these and we'll end the session. So please be interactive and try to guess or try to think of the answers. Next question is also related to DDT. Why was DDT banned? This is the question. Next question. Why was DDT banned? First option, it was expensive. Second option, it was heavy. Third, it was impossible. And fourth, it caused cancer. Now type your answers in the comment box. I'll wait for a minute. I'll repeat the question again. Why was DDT banned? You can also see it on my screen. Was it expensive? Was it heavy? Was it impossible to synthesize? Or did it cause cancer? Come on, there are 93 students. Please try to answer. See, this session is meant to be interactive. And the reason I'm telling you to try at least post the answers the comment box is also giving me a track of how many of you are really participating and listening to this session, right? And it will help you solving the assignments. Because if we turn on the mic for 100 participants, then it will be a chaos. Yeah, now there are the number of answers are increasing. Yeah, it's a follow up easy questions. We have already learned about what is DET and how does it do, how does it do its function, and almost all the people, almost definitely all the people, 100% have answered the correct answer. It caused cancer. So we rule out all the other possible options. It was expensive. No, it was not expensive because it's synthesized in a mass industrial process. So it was easily available to the farmers to use it on the field. It was not expensive. It was heavy. it wasn't heavy and it was impossible to synthesize. We already in the previous slide we saw there is a reaction, Friedel-Crafts reaction, in which these two, chloral and chlorobenzene, in the presence of an acidic catalyst, can be easily synthesized. DDT can be easily synthesized. And the fourth option is the correct answer. It causes cancer. Now let's see how DDT or where are the reports where DDT causes cancer. So what I'm showing you here is a title of a research paper or a research extract article published in Toxicological Research, official journal of Korean Society of Toxicology. So this is a published research article which they have verified their findings with experiments and supported the data. Title of the paper says Toxicity and Carcinogenicity of Dichlorodiphenyl Trichloroethane DDT, and this was published in 2015. So which is not even a decade back. It's like almost 6 to 7 years. Toxicity and carcinogenicity. So it says that DDT is toxic, carcinogenic. So by the term carcinogenic, I mean cancerous. Carcinogen is a agent which can cause cancer or which can induce tumors in the body. Is known as carcinogen. So this lab or research group have published in toxicological research about toxicity and carcinogenicity of DDT. Similarly, another research article here published in Public Health Research Perspective 2014. This is also not very old, 9 to 10, not even a decade old. Exposure to dichlorodiphenyl trichloroethane or DDT and the risk of breast cancer. A systematic review and meta-analysis. So this also shows that dichlorodiphenyl trichloroethane exposure increases the risk of breast cancer, and they have published their reviews and supported them with the experiment. Here is a scan showing increased risk of breast cancer in DDT exposed patients. So patients which were exposed to DDT in whichever form, may it be as a pesticide in the crop, which has incorporated in the crop, or via any forms, they have shown increase in breast cancer risk. Patients untreated with DDT, unexposed to DDT, and other people exposed to DDT have higher risk of getting breast cancer. And this is supported by two other well researched articles, toxicity and carcinogenicity of DDT. Now again we go back to the Stockholm Convention on Persistent Organic Pollutants. This is the same meeting which banned endosulfan in 2011. So Stockholm Convention laid down some rules, putting a global ban on several persistent organic pollutants and restricted DDT used to vector control. So what this convention did? It put a ban on DDT as an organic because it's an organic pollutant in the persistent organic pollutant in the field. Participants, please try to mute your mic and turn off your video chat while we are in the session. Yeah, because only then the session can proceed smoothly. So this Stockholm Convention put a ban on DDT use and restricted DDT use to vector control. So let me give you a side information of what is a vector control. DDT, it was seen that DDT use was very effective for eradicating malaria, and this DDT was also toxic to mosquito and larvae, which was an accidental side effect of using DDT. So when DDT was sprayed onto the fields, then the soil and water all contained DDT, and this water used to wash out to a pond or a pool, and this water stagnate there. Now we know mosquitoes love to lay eggs in the stagnant water. Mosquito laid eggs in the stagnant water where this DDT washed fertilizers or water, crop water was stored. Now what happened? These mosquito larvae incorporated DDT, and the same thing happened to them. Mosquitoes are no different than other arthropods or insects or pests. So their system is also similar. DDT went inside the mosquito larva and then sat on the ion channels, sodium ion channels, which caused constant firing of all the neurons in the mosquito larva, and the larva died. So what happened in DDT treated areas, or in DDT areas which used DDT as a fertilizer? In those areas, the birth of mosquitoes or the mosquito population, malaria population, was under control. So people suffered less from malaria because mosquitoes were dying, they were not able to lay eggs. So what this Stockholm Convention did, they laid down rules that DDT can only be used for eradicating mosquitoes, no longer as a pesticide for organic farming or any kind of farming, because it's a pollutant, and it's very cancerous because of all those studies. People cannot consume crops treated with the DDT, and it was effect in 2004. DDT was banned, but DDT is still in use, like in backward places and others who do not have much information on this, and also illegally. But DDT is widely used as a vector control or controlling mosquito population and fly population. So DDT was banned. Now let us look at how DDT is uh, becoming toxic. How did it reach humans? So as we have read about bioaccumulation, that it accumulates in the organism in case of endosulfan, what happens in DDT is another phenomenon called biomagnifications. Now, biomagnification is a phenomena in which higher amount of toxins are present as trophic level increases in the food chain. So here is a depiction. If you just don't look at this complex diagram for now, let's go with me. This is a trophic level diagram. For students who do not know what is a trophic level diagram, here this just depicts the order of organism in a food chain. So humans as the top consumers, we are consumers and the plants are producers. Humans as consumers sit on the top of the trophic table, and algae are the producers. Which green algae produces food by photosynthesis, and they are the producers. They fix energy. Now, and there are other consumers and producers in between these trophic levels, and this is a total food chain. So if you see, green algae is the bottommost producer. Then there are something called zooplankton. Zooplankton are organisms, very minute microorganisms living in the sea and ponds. What they do, they feed on the algae. So they will come and eat the algae, zooplankton. The next trophic level is occupied by small fish. These small fish will now eat the zooplankton. So there are some fishes which feed on this zooplankton and they survive on this. Then comes the next trophic level, which is the large fish. Large fishes will eat these small fishes. Large fishes' food is the small food. And humans will fish. We do fishing, all sorts of fishing in the pond, in the lake, river, seas, and oceans. We will catch this large fish and we will consume it. So now what does biomagnification says? So if there is a DDT compound or a DDT presence in the algae, as we move higher in the trophic table, from algae to zooplankton to small fish to large fishes and humans, the level of DDT will magnify with each trophic level. So if you see, there are two crosses in the algae here. Now in the zooplankton, there are many crosses. And small fish, many more. And large fish, many more. And humans, there are lots of. So these crosses are just a depiction of DDT concentration. And this blue-black thing legend here says amount of toxins, parts per million. So here is a calculative picture of how much toxin the algae has. Let's say if the algae, which is growing in the sea or in the pond, gets polluted water with the DDT and it incorporates it, 0.04 parts per million of DDT in the algae. Now the zooplankton will eat this algae, will feed on this algae, their DDT concentration will magnify to 0.23 parts per million. You see, there is a huge change. 0.04, now 0.23. This might seem very insignificant, but if you see, there is a tenfold increase because the decimal has the numbers have moved 10 place to the decimal. Tenfold increase in the zooplankton. Now, zooplankton will be the food for small fish. Small fish will feed on the zooplankton. Zooplankton had 0.23 parts per million DDT, but the small fish now has more accumulation of more magnification of DDT. So it's just not accumulation; it's increasing in DDT concentration, 2.07 parts per million. So it's again it's a 10 time fold increase. So in each trophic level, as we move over to the trophic level, then again the large fish will eat the small fish. Now we have again drastic increase in the biomagnification of the DDT, and then large fish will finally reach the humans, 13.55 parts per million of DDT in humans. That's why the DDT even is considered so toxic. If the DDT enters the lower trophic level or algae and plants, then it can biomagnify till it reaches the humans with with various trophic levels. That's why DDT is so dangerous. Endosulfan, on the other hand, it was accumulated in the organism. It will pass on down to the generations of the organism and then stay in the same amount in the body or what in the consumer. But DDT magnifies, and this happens because see algae are in more number. If you see size wise, algae are very small. Zooplankton little bigger than algae but still small. Small fish greater than zooplankton but still small. So in comparison to the upper trophic level, the sizes of the organism are increasing. Humans are the largest. Large fish is still small in comparison to humans and all. So the amount of feeding this zooplankton will do on algae is more. So if a single algae contains 0.04 parts per million, the zooplankton will eat more of these algae and accumulate more of this. Then several zooplankton will be eaten by small fish, several small fishes will be eaten by large fish, and humans will be consuming large fishes all the time. Even humans can also consume small fish and small fish and take their DDT. So this is how upon the feeding trophic zone or the trophic levels, the biomagnification phenomenon is seen in case of DDT. That's what makes DDT very dangerous. That's why DDT was banned in way back in 2004, even earlier than endosulfan, because it's too dangerous and will cause these neurons to fire and spams and eventually death. It can cause death of pests, but it should not be used because it can reach humans through biomagnification. Is it clear to the students? Is there any difficulty in understanding why DDT is banned? And the answer that we found out was it causes cancer and other biomagnification attributes. Is it clear to the students? Just mention in the comment box. Okay, okay. Yeah, yes, got it. Yes. Abhishek, Rabisha, Bikram, Tara, Yashwant, Somideep. Yes, okay. Good that you guys are trying to keep up and able to follow. I have tried to simplify things for you so that you understand what is the reason behind the answer. Because anyway, what questions what we are seeing in the assignment and in this practice assignment also are just MCQ questions. You can just click one answer and then just go forward. But what we need to see here is logic behind why we choose that answer and what is the story behind it, so that whenever we encounter another form of the same question regarding the same topic, we can answer it. So now I'm very confident that any question regarding DDT, if it comes across your assignment or in any part of your course, and you will be able to encounter it. Am I right? If there can be, now you know all about DDT, at least briefly. If there will be questions on DDT, then you will be answer. Am I right? Will you be able to answer, students? You can just write yes or no. So this is just a method for me to assess that you are actually absorbing what we are discussing in these sessions. Yes, yes, it makes me happy to see that you guys are understanding. Very good. So now moving on to the last two questions. It's almost time. Let's quickly find out this answer. What part of plant does rhizobium mostly forms nodule at? So it's a completely different kind of question from the previous ones where we are encountering pesticides and insecticides. What part of plant does rhizobium mostly forms nodule at? The options: leaves of the plant, flower, stem of the plant, roots. I'll give a minute for all the students to just answer. See, students don't feel shy. Please try to answer. It's okay if your answer is not matching, but the attempt, the effort is necessary. And we will all find out what is the right answer and what is the reason behind, what is the rationale behind the answer. I'll repeat the question. What part of plant does rhizobium mostly forms nodule at? Leaves, flower, stem, or roots? I'll wait for a minute. Some people have already typed their answers. Others, please type their answers in the chat box. Now let's see what's the correct answer. The correct answer is roots. Oh, seems like you guys all knew this answer. It was an easy question for you. Good, very good. The correct answer is roots. Now we will see what is rhizobium and what kind of plant does it form this nodule or whatever in the roots. Now here you see a diagram of a plant. This is a certain kind of plant called a leguminous plant. This is a plant belonging to a certain family called a legume family, or leguminosa or fabaceae. And only in these roots, the roots of these kind of plants of this family of leguminous plants, root nodules form, harboring the rhizobium bacteria. So rhizobium here is a bacteria. It's a very famous bacteria, very well known. It is the kind of bacteria which can fix nitrogen, atmospheric nitrogen. Remember Haber's Bosch process? Fixing atmospheric nitrogen even into ammonia or reacting atmospheric nitrogen anywhere is very difficult. Even plants cannot fix atmospheric nitrogen themselves. So some leguminous plants have have devised this crazy idea where they are very clever. So the plants know they cannot fix the nitrogen for themselves. So what they do, they go and befriend the bacteria which can fix the nitrogen. So there are these rhizobium bacteria which are able to fix atmospheric nitrogen. These plants have come into a symbiosis agreement or a mutual deal or arrangement. The plant will say to the bacteria, you fix nitrogen for me, and in return, I will provide you shelter and other nutrients. So the bacteria readily agreed. That's how the symbiosis relationship exists. Root nodules of leguminous plants harboring the rhizobium bacteria. In this diagram, we see in this nodules, bacterium fix nitrogen from air, and the plant fixes photosynthesis products. Plant can photosynthesize, but certain bacteria cannot. So photosynthesis product means all these metabolites, glucose, and other ions, these plants provide these bacteria these ions. And this kind of relationship, as I have mentioned earlier, is known as symbiosis. So symbiosis is a relationship of mutual benefit of two interacting partners. So if you see what is a symbiosis, any two in any two partners, they can be of the same species, different species. So in a symbiosis relationship of nodule, we see a different species, interspecies symbiosis, where a species of bacteria symbiosis has a symbiosis relation with a leguminous plant. So a relationship of mutual benefit. So the bacteria is benefiting out of this relationship, and the plant is also benefiting out of this relationship. The plant is gaining nitrogen and bacteria is gaining photosynthesis products. So if we draw this schematic, so one of the partners, interacting partners, is a leguminous plant, or this plant, and the other partner is a rhizobium bacteria, and both mutually benefit. That's why it's called a symbiosis relationship. So here is a schematic for leguminous plant, and this is how it looks like. Under the ground, the root hairs or the root extensions have bulge-like swelling structures called root nodules. And if we magnify these root nodules or cut through them and see, these root nodules contain nitrogen-fixing bacteria like rhizobium, which can fix the atmospheric nitrogen into nitrogenous compounds. So these bacteria can take up atmospheric nitrogen and fix them into nitrogenous compounds, which is assimilable in the plants. So these plants can now take up these nitrogenous compound and use them. Earlier, the plant was not able to use atmospheric nitrogen. So what the bacteria did, it helped the plant. Okay, I will fix the nitrogen into some compounds, and then you take up the compound and you use it as a nitrogen source. In return, I will have shelter and I will take some of the photosynthesis product from me. This is what happened. Now let's take a look at how the nodule is formed, how a root nodule forms. It's a very simple, phenomena, and it is characterized into four stages. So first, this is a schematic for root hair in soil particle. So let's say this area of the root, very finest tip of the root, where we have root hair cells extension into the soil particles. Hello, yeah, is my voice audible? I thought I my presentation went down. Yes. Okay, okay, okay. Fine, fine. I'll just continue. Sorry. Yeah, as we were speaking. Yes, about the nodule formation, the process of nodule formation. So first, what happens? Root hair cells present around the soil. So here we show this is the root hair. Let me switch on my pointer. And there are some bacteria around in the soil. So we all know bacteria are very tiny organisms, and there are many kinds of bacteria present in the soil too. Some kind of bacteria, will try to accumulate near the root hair. Then the next step is these bacteria will secrete some factors like some proteins. Bacteria will secrete some factors, proteins, and which will cause the root hair to bend in like a hook like structure. So there is a morphological feature or a physical feature which is seen. The root hair turns into a hook like structure around the bacteria. So the bacteria is present here. The root hair will now curl around the bacteria and form a hook like structure. Now what happens in this hook like structure, which is surrounded by the bacteria? Why did it form a hook? The hook will provide lot of surface area for the bacteria to infect. Now the bacteria has a lot of access to the surface of the root hair, to infect. So now this rhizobium bacteria, one of the rhizobium bacteria will infect into the root hair. Now after one bacteria infects into the root hair, that bacteria will secrete some other attracting factors for its own bacteria, own kind. So all its friends will follow the first bacteria into the root hair. Now slowly these bacterium will form a chain which is called an infection thread containing all the bacteria, and this thread will grow along long and will go inside the root cortex. So inner cortex and pericycle cells under division. Root hair cells will be root hair cells have pericycle and cortex cells at the top. So this infection thread containing bacteria will move into through the root hair and move into the pericycle and cortex. Now in these pericycle and cortex cells, the bacteria will infect them and then they will live inside them and form their colonies as a mature nodule. So whatever we see a nodule is not the root hair cell but the pericycle and cortex cell which have been infected by the rhizobium bacteria. So this is how the root nodule formation happens. First the bacteria surrounds the root hair. Next it causes the root hair to form a hook kind of structure so that it can have lot of surface for infection. Third is infection formation of infection thread. The bacteria will secrete some attracting factors which will attract other bacteria inside the root hair, and then they will infect the inner cortex and pericycle cells under division, the fourth step, to form a mature nodule. This is the whole process of root nodule formation in leguminous plants. So to the answer of our ninth question, which all of the students answer correctly. So roots are the part of the plant in which rhizobium infect. Coming to the last question of this session, 10th question is also the question reads, which of these plants can produce fixed nitrogen themselves by symbiotic relation? Okay, see this is I will just ask all of you to quickly I'll just ask all of you to quickly answer this question. The options are wheat, peanuts, paddy, rice, and cabbage. So this is a follow-up question of what we just learned right now. Which of these plants can produce fixed nitrogen themselves by symbiotic relation? It's okay if you do not know the answer, you can just guess. I will give you a hint, or you might already have the hint. Whichever of these plants is a leguminous plant will be able to fix nitrogen themselves by symbiotic relation, right? Okay, there are some answers already. Option 2, peanuts, peanuts. Okay, peanuts. Lots of peanuts. I want to tell you, you all guys are correct. It's peanuts. Very nice. Congratulations. We have come to the end of our session. Just let's see what other kinds of legumes are there and why wheat, paddy, rice, and cabbage cannot be. Because this is the only leguminous plant. Wheat, paddy, rice, cabbage are not leguminous plants. These are cereal pulses. Paddy, rice, wheat are cereals and pulses. So legumes are of family. Legume is a plant family in the Fabaceae, in the family of Fabaceae, or also known as Leguminosae, or the fruit or seed of such a plant. And these are some of the examples of the legume family: beans, common bean, pea, lentils, peanuts, the answer that we had in the last question, broad bean, soybean, chickpea, mung bean. So you see all kinds of bean family and these pea family are all legumes. Now, uh, do you know the breakthrough of genetic studies done by Mendel, Gregor Mendel, used one of the legume plants as their model organism? Which of them is it? Can somebody answer? This is a bonus question. Which of the legume plant was a genetic model for discovery of genetics by Gregor Mendel? Very good. I'm so thrilled to see that students have all their basics cleared. You are all correct. It's the pea plant. Answers are mentioning peas. Very good. So it's the pea plant on which genetic experiments, the whole field of genetics was discovered by a great scientist Gregor Mendel. So legumes are also called pulses. When used as a dry grain, the seed is also called a pulse. So if you dry this pea, it will become yellow peas. These are pulses. Lentils, we have many South Indian delicacies and North Indian foods using lentils in dosa batter and everything. Chickpea is a common delicacy. Mung bean, broad bean, peanut, we all have. So these are all pulses when dried. And legumes have been cultivated way back in history since 70,000 years ago by Neanderthals. It was also found in Indus Valley civilization way back in 3300 before Christ. Egyptians also used lentils for their cooking, and dry pea seeds were also believed date back to the Stone Age. So this is just a history and short of legume. You have answered it all correctly. So with this, we have come to the end of this assignment solving session. I hope you guys could have understood what we have learned today. I'll stop presenting my screen and switch on my video chat. Yes, I hope all of you guys had a good understanding. It may not be the exact assignment that you are solving, but these kind of questions will help you to generate the thinking that you will need for solving these assignments. Now, if you guys have any quick questions which I missed, you can please ask me related to these assignments or any problem that you're facing with your assignments. See, if you ask questions like when will the solution come and week 2, week 1, I seriously have no idea because I'm here to help you with the assignment. I'm a teaching assistant. So you can mail Professor Mainak Das or the NPTEL team to inquire when the scores will be out or when the correct answers will be displayed. If you have any question regarding to what we learned today, of all the 10 questions we solved, or any concept in your weekly lectures, I hope you must have gone through it. If not, please go through it. And it's already 5. So every Sunday we'll be having this practice assignment week. So today was week 1, first week related to like the first assignment week 1. Okay, no problem, Tara. You can always solve the upcoming assignments. If you miss 1 2 assignments due to late registration and others, I think that won't be considered, or your performance in the other quizzes will be absolute. You can talk to the course instructor, Professor Mainak. I just want to tell you, student, that you have done pretty well in interaction in this problem solving and interaction session. The questions were easy, but we tried to understand what is the rationale behind these answers. And every Sunday we will be having corresponding problem assignment solving where I will help you guys with such interactive sessions. So with this, I'll end this session. Thank you for joining. Thank you for your time. Yes, 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 Vikram. You have a question? Yes, Varsha, this session will be recorded and uploaded. So you will receive some details. My YouTube channel will have these recorded sessions, and I'll also upload these slides which I have prepared, which will be provided to you by the NPTEL coordinator. So for all the students who can access these slides and the recorded session. So I'm already recording these sessions and it will be available on my YouTube channel. And the YouTube channel link will be sent to you communicated to you via the NPTEL coordinator once I fill in my details. Okay. Thank you for joining in the session. Thank you.